Hello everyone, this is Dr. P. Mary Anupama, Assistant Professor and Head of the Department of Biochemistry, St. Joseph's College for Women. So the topic that we'll be studying is TCA cycle and we'll be studying about its regulation. The contents that will be covered include the conversion of pyruvate to acetate, then introduction to citric acid cycle, the reactions of TCA cycle, then regeneration of oxaloacetate, energetics of citric acid cycle, inhibitors of this cycle as well as regulation. Conversion of pyruvate to acetate. We all know that pyruvate is the end product of glycolysis and that is converted to acetyl-CoA by oxidative decarboxylation reaction. So this reaction, it is considered to be irreversible and is catalyzed by a multi-enzyme complex. So this multi-enzyme complex is nothing but the pyruvate dehydrogenase complex, and this is found only in the mitochondria. This particular multi-enzyme complex, pyruvate dehydrogenase, it requires 5 cofactors, or we call it as 5 coenzymes. These include TPP, thymine pyrophosphate, lipomide, FAD, coenzyme Q, as well as NAD+. So here, lipomide, it contains lipoic acid, which is linked to the epsilon amino group of lysine residue, and it is required for the reaction. So you see here the conversion, pyruvate is converted to acetyl-CoA in the presence of pyruvate dehydrogenase complex. NAD+ is required, coenzyme A, and it is converted to acetyl-CoA. One molecule of carbon dioxide is released and NADH+, H+ is produced. Citric acid cycle, it's also called as Kreb cycle or tricarboxylic acid cycle, or simply we call it as TCA cycle. It is one of the most important metabolic pathways which is required for supplying energy to the body. Around 65 to 70% of ATP, it is produced through this pathway only. And this is the pathway which is very important for oxidation of acetyl-CoA to produce carbon dioxide and water. And this cycle, it utilizes 2/3 of total oxygen which is consumed by the body. 2/3 of oxygen. And why is it named as tricarboxylic acid cycle? It is because it results in the formation of citrate, cis-aconitate, and isocitrate, which are tricarboxylic acids. As it results in the formation of these three, we call it as tricarboxylic acid cycle. And this cycle, it is also named Kreb cycle after the person who has proposed it. You know that it is proposed by Hans Adolf Krebs in the year 1937. Hence, we call it as Krebs cycle. He is the person who has studied this Krebs cycle. He has observed oxygen consumption in the Phrygian breast muscle. And for this study of his, he was awarded Nobel Prize for Physiology and Medicine in the year 1953. Citric acid cycle, it totally comprises of 10 steps. The first step is the formation of citrate. 2 and 3 steps will result in the formation of isocitrate. Citrate is isomerized to isocitrate in 2 and 3 steps. In the steps 4 and 5, it results in the formation of alpha ketoglutarate. The step 6 is conversion of alpha ketoglutarate to succinyl-CoA. 7th step is formation of succinate. 8th step is conversion of succinate to fumarate. 9th step is formation of malate, while the last step, 10th step, is conversion of malate to oxaloacetate. Regeneration of oxaloacetate takes place here. So these are the 10 steps of citric acid cycle. Let us see each of them in detail. First step is formation of citrate. This is a condensation step in which oxaloacetate condenses with acetyl-CoA, resulting in the formation of citrate molecule. The citrate or the citric acid is produced and the reaction is catalyzed by citrate synthase. In this you can see that coenzyme A is released from the acetyl-CoA, while these two condense to result in the formation of citrate molecule, the first tricarboxylic acid molecule to be produced in the citric acid cycle. The steps 2 and 3, these involve isomerization of citrate to isocitrate. This reaction, it is catalyzed by aconitase. During this, this is a two-step process. Here we have represented single step, but an intermediate will be there, which is cis-aconitate. So this is a two-stage conversion wherein citrate is converted to isocitrate by aconitase, while the intermediate is cis-aconitate. So this is the 2 and 3 steps of citric acid cycle. Steps 4 and 5, they result in the formation of alpha ketoglutarate. The enzyme that is involved in this is isocitrate dehydrogenase, and it catalyzes oxidative decarboxylation of isocitrate to alpha ketoglutarate. Isocitrate to alpha ketoglutarate. So here again it is a two-step process. An intermediate is produced over here, which is oxalosuccinate. So isocitrate results in the formation of oxalosuccinate and then alpha ketoglutarate. This reaction is also accompanied by the formation of NADH, and there is a liberation of carbon dioxide molecule. Isocitrate is converted to alpha ketoglutarate finally through an intermediate which you call it as oxalosuccinate. Sixth step is conversion of alpha ketoglutarate to succinyl-CoA. This again occurs through oxidative decarboxylation, and the enzyme that is involved is alpha ketoglutarate dehydrogenase complex. So this multi-enzyme complex, it requires again cofactors for its activity, which include the TPP, thymine pyrophosphate, lipomide, NAD+, FAD, as well as coenzyme A. The mechanism of conversion, it is very similar to that which we have studied previously, catalyzed by pyruvate dehydrogenase. Pyruvate dehydrogenase is also a multi-enzyme complex. In this step again, another molecule of NADH is produced, accompanied by release of one more molecule of carbon dioxide. So this is the second carbon dioxide molecule which is released. Seventh step is formation of succinate. So succinyl-CoA, which is produced here from alpha ketoglutarate, it is converted to succinate by the enzyme succinyl-CoA synthase. So here, in this reaction, you see one phosphorylation taking place wherein GDP is phosphorylated to GTP. So this we call it as a substrate level phosphorylation step. Okay, and this GTP is later on converted to ATP, and the enzyme that does it is nucleoside diphosphate kinase, and the coenzyme A, it is released off. So finally you have succinyl CoA getting converted to succinate by the enzyme succinyl CoA synthetase. Eighth step is conversion of succinate to fumarate. So the succinate, it is oxidized to fumarate, and the enzyme that is involved is succinate dehydrogenase. In this reaction actually there is production of FADH2. FADH2 is produced. Ninth step is formation of malate. From the fumarate you are getting malate, and here one molecule of water is added to fumarate, and the enzyme that does this reaction is fumarase. Fumarase converts fumarate to malate. And the last step is conversion of malate to oxaloacetate. So malate, it is again oxidized here to oxaloacetate by the enzyme malate dehydrogenase. In this step, the third and the final molecule of NADH is produced. And this is the step which regenerates oxaloacetate, so that this oxaloacetate will combine with another molecule of acetyl CoA and will result in the molecule in formation of citrate, and the cycle continues. So oxaloacetate in the first step has condensed with acetyl CoA to form citric acid. Okay, while in the 10th step it is oxaloacetate which is produced, which is ready for another condensation reaction with acetyl CoA to form citrate. So here is the summary of the cycle. Oxaloacetate condenses with acetyl CoA to produce citrate catalyzed by the enzyme citrate synthase. Now this citrate is converted to isocitrate by the enzyme aconitase. This is a two-step process. Isocitrate is converted to alpha ketoglutarate by the enzyme isocitrate dehydrogenase. This is again a two-step process, and here you have NADH produced and one molecule of carbon dioxide is lost. Alpha ketoglutarate is converted to succinyl CoA, and in this step you can see NADH is produced and another molecule of carbon dioxide is lost. The reaction is catalyzed by alpha ketoglutarate dehydrogenase. Now the succinyl CoA is converted to succinate by the enzyme succinyl CoA synthase. Here it is a substrate level phosphorylation resulting in the formation of GTP. Coenzyme A is released. The GTP is converted back to ATP. Succinate is converted to fumarate, and here you have FADH2 produced, which enters the electron transport chain. Now the fumarate, it is converted to malate. One molecule of H2O is added to fumarate by the enzyme fumarase. Now malate is produced. This malate is now converted to oxaloacetate, wherein you have one more molecule of NADH is produced. The reaction is catalyzed by malate dehydrogenase. Now this oxaloacetate is ready to enter another sequence of events. So this is how citric acid cycle operates. Here is the summary of TCA cycle, wherein acetyl CoA, which is produced from pyruvate, it is completely converted to carbon dioxide. This is a 2-carbon molecule and 2 molecules of carbon dioxide is produced. So this will result in the formation of 3 molecules of NADH, 1 molecule of FADH2 and 1 GTP molecule. Okay, and you see that the carbon is totally oxidized to carbon dioxide. The GTP which is produced is again converted to ATP. Then inhibitors of Krebs cycle. So there are some inhibitors, important ones of TCA cycle, which are presented in this table. So the enzyme and inhibitor are mentioned over here. Here it is aconitase and the inhibitor is fluoroacetate, which is a non-competitive inhibitor. Next we have alpha ketoglutarate dehydrogenase, which is inhibited by arsenate, a non-competitive inhibitor, and succinate dehydrogenase, which is inhibited by malonate, which is a competitive inhibitor. So here fluoroacetate, it is actually initially it is activated to fluoroacetyl-CoA. Okay, this fluoroacetyl-CoA, just like how acetyl-CoA has condensed with oxaloacetate, this will go and condense with oxaloacetate and will result in the formation of fluorocitrate. And that fluorocitrate, from fluoroacetate you get fluorocitrate. And this goes and inhibits the enzyme acetyl-CoA, aconitase. So this reaction is considered to be suicidal. Once fluoroacetate is converted to fluorocitrate, that's it. It goes and binds there and aconitase is not available for the enzymes to act. Regulation of citric acid cycle. The regulation of citric acid cycle is because of many different enzymes, and also it could be at the level of ADP. The three important enzymes that will be regulating its activity are the citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase. What does this citrate synthase do? Here you see that citrate synthase, it is inhibited by ATP which is formed, and even NADH, even acetyl-CoA and succinyl-CoA. So if excess of it is there, it will be inhibited. Next is isocitrate dehydrogenase. So isocitrate dehydrogenase, it is activated by ADP. If ATP is less, ADP will be more. So isocitrate dehydrogenase activity is activated by this, and it is inhibited by ATP and NADH. Third one is alpha ketoglutarate dehydrogenase. This particular enzyme, it is inhibited by succinyl coenzyme A as well as NADH. So why is availability of ADP very important here? You see that citric acid cycle, if it has to proceed, there should be less of ADP. So if insufficient amounts of ADP is there, then only it will take place. And even oxidation of NADH and FADH2 through the electron transport chain, if it stops if ADP is not there, because in electron transport chain as they enter, they will be resulting in the formation of ATP. Okay, so if this is happening, it will lead to accumulation of the molecules and the molecules which require them. So obviously it will be inhibiting the enzymes that require NAD plus and FAD. So both of these, they are very essential for the TCA cycle to proceed. So if ADP is insufficient, then all the cycle stops. Amphibolic nature of citric acid cycle. So this citric acid cycle, it provides many intermediates for the synthesis of many compounds which are required in the body. The Krebs cycle, it is considered to be both catabolic as well as anabolic in nature, and hence it is also called as amphibolic. And this TCA cycle, it is involved in the products of it. They are involved in gluconeogenesis, in the transformations as well as deamination reactions. You have many anabolic reactions also, which take place because of the citric acid cycle. Okay, so here you can see citric acid cycle. Acetyl-CoA, it's getting converted to citrate. From the citrate, you get fatty acids and steroids. Alpha-ketoglutarate. So you see that it is the product of transamination of glutamate. So there is interconversion between glutamate and alpha-ketoglutarate. And this glutamate, it is required for the synthesis of non-essential amino acids as well as purines. Next one is succinyl-CoA. It is required for the synthesis of heme. Malate. Malate can be converted to pyruvate. Okay, this is again reversible. Oxaloacetate is converted to pyruvate. Okay, then you can see oxaloacetate. It is also undergoing transamination reaction to produce aspartate. So aspartate is again required for the synthesis of some amino acids, purines as well as pyrimidines. That's why we call it as thanoplerotic pathways. All these we call them as thanoplerotic pathways, which are utilizing the intermediates of citric acid cycle. Let us discuss some important anaplerotic reactions, of which the first one is formation of oxaloacetate. So pyruvate carboxylase, it catalyzes the conversion of pyruvate to oxaloacetate, and this reaction is ATP dependent, and this oxaloacetate enters the citric acid cycle. So pyruvate, it is also converted to malate by this NAD plus dependent malate dehydrogenase. So pyruvate is converted to oxaloacetate by the enzyme malate dehydrogenase. Next is the important one, transamination. So wherein it's a process where the amino acids transfer the amino group, the amino group to, so there is one keto acid. The keto acid will accept the amino group from another amino acid and it will become amino acid. Okay, while amino acid which has contributed the amino group will become the keto acid. So it is an interconversion. So it's the same like alpha ketoglutarate to glutarate. While you can see here formation of alpha ketoglutarate as well as oxaloacetate is because of the transamination. So alpha ketoglutarate synthesis to glutamate, it is taking place because of the enzyme glutamate dehydrogenase. You can see here glutamate is getting converted to alpha ketoglutarate and NADP plus is involved, resulting in the formation of NADPH plus H plus, and amino group is withdrawn from the glutamate, which will be transferred to another amino acid. Energetics of glucose oxidation. So glucose molecule as such is a 6-carbon molecule, a hexose molecule, and during glycolysis we have seen that it has resulted in the formation of 2 pyruvate or 2 lactate molecules. Both of them are 3-carbon molecules. The pyruvate molecule, it is initially oxidatively decarboxylated to result in the formation of oxaloacetate, acetyl-CoA. Pyruvate is oxidatively decarboxylated to result in the formation of acetyl-CoA. This acetyl-CoA condenses with oxaloacetate to form citrate. That is the first step. So this alone enters the citric acid cycle, and it's a 2-carbon molecule wherein it is completely oxidized to carbon dioxide by the end of this cycle. So the overall process of glucose oxidation to carbon dioxide and water through glycolysis and citric acid cycle, all of this is represented in this equation. You can see C6H12O6 plus 6 oxygen molecules in the presence of 38 ADP and 38 inorganic phosphate. All these 38 ADP, they are phosphorylated to form 38 ATP molecules, while the glucose is completely oxidized to carbon dioxide and water. When you use the instrument colorimeter and when you start burning the glucose, it will result in liberation of 2750 kilojoules of heat. Okay. So in the living systems, the energy is stored in ATP. So 38 ATP are produced in the living systems when glucose is oxidized, and this is equal to 1159 kilojoules. So this accounts to around 48% of energy which the glucose has in it. The textbooks that are referred while preparing this material, it is Principles of Biochemistry by Leninger, Biochemistry by U. Satyanarayana, as well as Biochemistry of Harper's. So the topics that we have dealt here, it is conversion of pyruvate to acetate, followed by acetate entry into citric acid cycle, all the reactions of the TCA cycle we have seen, then regeneration of oxaloacetate we have discussed, then energetics of citric acid cycle we have discussed, and then inhibitors, and then regulation. All these are the topics we have covered. Thank you.